lead back to spike protein, right? So COVID is a bizarre disease, right? It does a lot of damage to a lot of different systems, as Pierre can tell you. The fact that the vaccines utilize spike protein at the level of the drawing board makes sense. But this was done at the drawing board before it was understood that the spike protein itself was cytotoxic. Now, one of the things that we got tremendous pushback for on my podcast with Robert Malone and Steve Kirsch was the claim made by Robert that spike protein is cytotoxic. Cytotoxic means kills cells. This is actually unambiguous, and the pushback was actually very carefully phrased, because what they're really saying is that the spike protein in the vaccines is not cytotoxic as far as we know. There's no evidence of that, right? But as Robert points out, this is nonsense, because what we know, what we learned too late to prevent the vaccine manufacturers from using spike protein, was that spike protein is cytotoxic, and the subunit that they have used is based on that spike protein. Now, they have locked it, so this is a protein that changes form, right? It basically closes like a clamp, and they have modified the sequence to lock it open in order that the part of the spike protein that is, this is too deep in the weeds probably, but that is not covered by sugars, right, is available for the immune system to discover it. So they've locked it open. And there is a possibility that that would prevent it from being toxic. But they didn't design it to be non-toxic. They locked it so that the immune system could see it. And the problem is that this vaccine, or these vaccines, have already failed at several different levels. The way the vaccine is supposed to work, it is supposed to be injected into you. At the injection site, it is supposed to have the mRNAs or the DNA enter the cells, trigger the production of spike protein. The spike protein is supposed to move to the surface of the cell, and it is supposed to stay there. It has a domain in it that is supposed to stick it into the cell surface where the immune system is supposed to see it and learn it, right? Now, the fact is, the components of the vaccine do not stay in the injection site, and the spike proteins do not stay locked to the cell surface. Maybe some of them do, but many of them seem to float around the body. So we have this molecule, which is based on a COVID molecule or a SARS-CoV-2 molecule that is cytotoxic, that circulates around the body. The evidence is that it actually shreds the blood-brain barrier, so it opens up holes into the brain. So, when you're talking about long COVID, post-vaccine syndrome, and these people have brain fog and other cognitive disabilities, it makes sense that there's been an error here, right? You've got a spike protein capable of damaging this tissue necessary to protect the brain. The spike protein seems to be circulating around the body in a way that the designers of the vaccine did not intend it to do. And so it all sort of adds up that COVID itself, long COVID, after the virus is gone, but there are still viral proteins, probably spike protein, and post-vaccine syndrome, where the spike protein has been produced in isolation of the virus, all of them have a similar collection of symptoms. Great. And this would also explain why ivermectin, whatever its mechanism of action, and there seem to be several, seems to be effective in treating all of them. But it's all telling us a kind of remarkable story. And, you have to ask, if you put the question to a business school class, what would you expect the behavior of a corporation that manufactures a product to be at the point you've immunized them from liability, right? Mm-hmm. I think the answer would be obvious, right? You would expect them to become a lot less sensitive to the harm that their product does and to pursue profit in spite of potential harm. Is that what's going on? Because it sure seems logical that the behavior would come from that calculation.
Now, let me ask you this about the spike protein. This effect, first of all, how do we know that it's not staying in the area of the injection, going throughout all the body and crossing the blood-brain barrier? How has this been measured? And why